Scientists have made a groundbreaking discovery regarding the phenomenon of virgin birth, unlocking the hidden mechanism behind this extraordinary occurrence. Furthermore, they have successfully activated this ability in female flies, shedding light on the fascinating world of reproductive biology. For the very first time, scientists have successfully identified a genetic explanation for the phenomenon known as virgin birth. This extraordinary ability, once activated, has the remarkable capacity to be inherited by subsequent generations of females. In a groundbreaking achievement, researchers have successfully triggered parthenogenesis, or virgin birth, in a species of fruit fly known for its sexual reproduction, the Drosophila melanogaster. This capability, once triggered in the fruit fly, can be inherited by subsequent generations. The descendants have the capacity to reproduce through sexual means if males are present, or through parthenogenesis if males are absent. Sexual reproduction is the predominant method of reproduction in the animal kingdom, wherein the fertilization of a female's egg occurs through the introduction of a male's sperm. However, there exists a phenomenon known as virgin birth, or parthenogenesis, wherein an egg develops into an embryo without the need for fertilization by sperm, thereby obviating the requirement of a male presence. Offspring resulting from a virgin birth do not possess identical genetic makeup as their mother. However, they exhibit a strong resemblance and are consistently of the female sex. Dr. Alexis Sperling, a researcher at the University of Cambridge and the primary author of the paper, expressed their enthusiasm about the groundbreaking discovery. They stated that their research has conclusively demonstrated the feasibility of manipulating the reproductive process in animals to enable virgin births. Witnessing a virgin fly successfully generate an embryo that can mature into adulthood and then repeat the phenomenon was an exhilarating achievement. According to her, in our genetically modified flies, the female specimens displayed a behavior where they spent half of their lifespan, approximately 40 days, in search of a suitable mate. However, they eventually abandoned this pursuit and resorted to reproducing asexually. During the course of the experiments, it was observed that a mere 1–2% of the female flies from the second generation who possessed the capability of parthenogenesis were able to generate offspring. This phenomenon exclusively took place when there was a complete absence of male flies. Conversely, when male flies were present, the females engaged in the conventional process of mating and reproduction. In certain circumstances, the adoption of a virgin birth can serve as a tactic for survival. A singular occurrence of virgin births can serve as a means to ensure the continuity of a species. Today, the research findings are made available to the public through the publication of the study in the esteemed journal, Current Biology. In order to obtain their findings, scientists initially conducted a sequencing of the genetic makeup of two distinct variations of a different fruit fly species known as Drosophila mercatorum. While one variation relies on males for reproduction, the other exclusively reproduces through asexual means. By analyzing the genetic activity during the instances of fatherless reproduction, they were able to identify the specific genes that were either activated or deactivated. After identifying the candidate genes responsible for the phenomenon of virgin birth in Drosophila mercatorum, the researchers proceeded to modify what they believed to be the equivalent genes in the model organism Drosophila melanogaster. The outcome was successful, as Drosophila melanogaster gained the unexpected capability for reproducing through virgin birth. Over the course of 6 years, a comprehensive study was conducted involving more than 220,000 fruit flies that had never mated before. The researchers attribute the success of this discovery to the utilization of Drosophila melanogaster as the experimental subject. They argue that conducting this study in any other animal would have posed significant challenges. The fly has served as the quintessential model organism for genetic research for over a century, and as a result, its genetic makeup is extensively comprehended. Recently, Sperling, who conducted this research in the Department of Genetics, has relocated to the Cambridge Crop Science Centre. There they will focus on studying crop pests and aspire to explore the growing prevalence of parthenogenesis in insects, specifically within pest species, in the future. Sperling expressed concern over the potential consequences of ongoing selection pressure for parthenogenesis in insect pests. It appears that this pressure may ultimately result in exclusive reproduction through virgin births. Such a scenario could pose a significant challenge for agriculture due to the fact that female insects would only produce female offspring, effectively doubling their capacity for proliferation. Some egg-laying creatures, such as birds, lizards, and snakes, have the ability to naturally transition to a form of reproduction that does not require males. However, it is uncommon for sexually reproducing animals to engage in virgin birth. This phenomenon is typically only observed in captive animals and occurs when a female has been isolated for an extended period of time and has little chance of finding a mate. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.